Okay, dear students, today we will see the thyroid hormone synthesis with this model. Okay, so the thyroid hormone synthesized by thyroid gland, and the thyroid gland contains follicles. So the follicle is a spherical structure. It is lined by follicular epithelial cells, and center we have lumen. The lumen is filled with colloid. Colloid is a gel-like substance that contains the thyroglobulin. This thyroglobulin protein is synthesized by the follicular epithelial cells. So let me explain the steps in the synthesis of the thyroid hormone. The important raw material for thyroid hormone is iodine and tyrosine. The first, the iodide in the blood is trapped by the follicular cell by sodium iodide import. So sodium and iodide both are transported, and from here, so this is the basal membrane of the follicular epithelial cell, and this iodide which is trapped, it enters into the colloid, and here the iodide is transported into the colloid for exchange chloride. An anion exchanger is there, and it is also called pendrin. Now this iodide which is transported into the colloid, when it reaches the apical surface of the follicular epithelial cell, it is oxidized. So the third step is the oxidation, that is iodide is oxidized into iodine, and this step is catalyzed by thyroid peroxidase. Now this iodine gets attached to the tyrosine, which is a component of the thyroglobulin, and it forms iodotyrosine. So monoiodotyrosine and diiodotyrosine is formed, and this step is called organification. So in organification, there is formation of monoiodotyrosine and diiodotyrosine, and the next step is the formation of T3 and T4. So T3 is when one monoiodotyrosine combines with diiodotyrosine, it forms T3, and two diiodotyrosine when combines, it forms T4. So this step is called coupling. So coupling is monoiodo with diiodo, diiodo with diiodo. So now ultimately monoiodo, diiodo, T3, T4, everything is formed and it is present in the colloid. So after this coupling reaction, this colloid is taken up by the follicular epithelial cell by the process of phagocytosis, and this creates a space in the colloid which is called resorption lacunae. So any follicle which is actively secreting will show resorption lacunae, a space in the colloid. Now this this trapped material is fused with the lysosome and get proteolized, and T3, T4 are released, and this T3, T4 diffuses into the blood because they are lipid soluble, and this monoiodo and diiodo go for deiodination, and tyrosine again it is recycled for the colloid synthesis. So to sum up, sodium iodide import, iodide chloride antiport, then oxidation, then organification, then coupling, then again endocytosis, then here proteolysis takes place, then diffusion of T3 and T4 into the blood vessel. Thank you.